Cells are the building blocks of all living organisms, but their sizes vary dramatically. Understanding these differences is essential for GCSE biology, and today we'll explore the relative sizes of cells, their components, and how to compare them using standard form. Let's start by considering the scale of cells. Most animal and plant cells are between 10 and 100 micrometers in size. To put this into perspective, 1 micrometer is 1/1,000,000th of a meter, or 10 to the power of -6 meters. For example, a typical human cell, such as a red blood cell, measures around 7 to 10 micrometers across. Plant cells, on the other hand, are often larger, averaging between 20 and 100 micrometers. Now, smaller than most cells are prokaryotic cells, such as bacteria. These usually range from 1 to 5 micrometers. Their small size is partly due to their simpler structure, lacking a nucleus and most of the organelles found in eukaryotic cells. Despite their size, prokaryotes play critical roles in ecosystems, including decomposing organic material and aiding digestion. To truly grasp the scale of cells and their components, we can also look at organelles. For instance, mitochondria, the site of energy production in cells, are roughly 0.5 to 1 micrometer in length. That means they are small enough to fit hundreds into a single cell, but large enough to see with an electron microscope. Ribosomes, the protein factories of cells, are even smaller, about 20 nanometers in diameter. A nanometer is 10 to the power of -9 meters, or 1/1000,000th of a meter. The smallest biological structures we consider are molecules like DNA. A DNA strand is about 2 nanometers in diameter, making it far smaller than organelles like mitochondria or even ribosomes. Despite its size, DNA carries the genetic instructions vital for life. Understanding these sizes is not just about memorizing numbers, it's also about comparing them. For example, an animal cell is about 10 micrometers, while a bacterium is 1 micrometer. This means a typical animal cell is roughly 10 times larger than a bacterium. Similarly, a mitochondrion at 1 micrometer is about 50 times larger than a ribosome at 20 nanometers. To help compare sizes, GCSE Biology often uses standard form, which simplifies working with very large or very small numbers. For instance, 1 micrometer can be written as 1 times 10 to the power of -6 meters, while 1 nanometer is 1 times 10 to the power of -9 meters. Using standard form makes calculations and comparisons straightforward and helps avoid errors. Another important tool is magnification. Light microscopes allow us to see cells and some organelles, while electron microscopes provide much higher magnification, enabling us to view smaller structures like ribosomes and DNA. The formula to calculate magnification is straightforward. Magnification equals image size over actual size. Let's take an example. Suppose an image of a cell measures 100 millimeters and the actual cell size is 10 micrometers. First, convert units so they match. 100 millimeters becomes 100,000 micrometers. Magnification equals 100,000 over 10, which equals 10,000. So the magnification is 10,000 times. To summarize, understanding cell sizes and their components involves recognizing their scale, comparing their dimensions, and using tools like standard form and magnification. These concepts form a foundation for studying biology at GCSE and beyond. To help with revision, I have put links in the description to must-have revision guides and flashcards that have helped students get to the higher grades with ease, so don't miss out. Thank you guys for watching this revision video. Don't forget to subscribe so I can continue to make these videos and help you guys even further.